Today we will discuss about how this glycan plays a role in the tumor progression and what are the uh, therapies that can be used to target these tumor cells using this glycan. So let us first understand how these glycans get modified in the case of solid tumors. So this is the example of the solid tumors that is on the top right hand side, and we can see that there are several glycans that are uh, only formed whenever there is a mutations in the healthy cells that leads to this cancer like phenotype. Some of the glycans that are been observed in the literature are this TN antigen, STN antigen, and this GD2. That and all these glycans are related. All these aberrant glycans are the markers for the cancer cells. That is, in the healthy cells, they are not expressed. These glycans are not expressed or expressed at a very minimum level. So this can be used to distinguish the cancer cells from the healthy cells. And in the case of this TN antigen, we can see more of this GalNAc monosaccharides, and in the case of STN we can see the GalNAc along with the sialic acid antisaccharides, and in the case of this GD2 we can see both the GalNAc, sialic acid, and galactose also. So what are the functions of these individuals uh, glycans? So let us start with this TN antigen. So this TN antigen interact with this galactin lectin with this macrophage at the galactin lectin. Through this galactin lectin, we can say this TN antigen interact with the macrophage and dendritic cells through this galactin lectins that are expressed on this macrophage and dendritic cells, and thereby it will promote this angiogenesis and also suppress the inflammation that is caused because of this aberrant growth of the cancer cells. And simultaneously this STN antigen and GD2 interact with the Siglec, Siglec that is present on the innate immune cells such as these natural killer cells or NK cells, and thereby it will this helps in the suppression of this uh, immune response, or in other terms that it promote the immune suppression, the interaction of this STN and GD2 with the Siglec. And this is known as this ITIM mediated cascade to create this anti-inflammation response. So these are some of the function of this aberrant glycosylation that are expressed on the cancer cells. Now we will understand what are the since we know the role of this aberrant glycosylation, so now we can target these glycan structures and use them, and use this approach to develop some of the anti-cancer therapy. So one of the therapies that are been focused on is on this glycan structure that is known as CA15-3, or it is related to this MUC61. So what happens so let us understand in the case of cancer cells, this CA15-3 glycan structure is more predominantly expressed, and what it does is it will suppress this T cell proliferation, or it will reduce the activation of the T cells that can be shown by the dashed arrow. And so it is essential to target this CA15-3 so that the immune response of the host does not get suppressed. So these are the strategies that are mentioned on the bottom right corner, that is CAR T cells and monoclonal antibodies, and these T cells with the BITE antibodies, that it has this single chain fragment, different single chain fragment, and also it has the antigen that can detect this MUC16 ED. So how they target is that in the case of CAR T cells, they use CD8 plus cells and induce them with this antigen that can detect this MUC16 ED, or that is this receptors, or this antigen is close to this MUC16 formation, so that these CAR T cells when interact with this MUC16 ED, it can act on this MUC16, which is just nearby to this MUC16 ED receptor. And similarly, when in the case of these T cells, they use single chain fragment, or they use two different receptors. First one is considered to target this MUC16 ED, and another one contains the receptors that is used to activate the T cells whenever this single chain fragment interact with this MUC16 ED. So one takes care of the intercellular signaling path, and another takes care of this MUC16 ED receptor detection and binding. So these are some of the strategies that are been working, so that after it binds to this MUC16 ED, it can help in shedding this CA125 from these cancer cells, so that the cancer cells will not be able to get benefited from this expression of this MUC16, such as this it will not help in tumor microenvironment development. Yeah, one thing I forgot to mention is that this SIGLEC also not only help in this immune suppression, but also help in the formation of this TME, that is tumor microenvironment. So let us come back to our main point right now, that these therapies are used to shed these MUC16 structures. That we can see that here it is the MUC16 is bind to this cancer cells. After shedding, it is removed from the cancer cells, so that the cancer cells get deprived of this MUC16s, and thereby will not be able to survive for a longer duration of time, as the functions such as angiogenesis and this TME formation get reduced, and it will not be able to get the nutrient supplies that is necessary for the survival of any cells. So that's all for this video. If you need to understand more about this glycan and pancreatic tumor in detail, you can go through the reference that has been provided in the description. Thank you for your time.